Welcome to the lecture about synapses. So we talked about how cells have this funny shape with all these protrusions and all these dendrites because they want to connect to each other, because that's like what they do. They connect to each other and pass messages to each other. Like this cell has all these dendrites, lots of fibers coming to it. But so how do they connect to each other? How do these connections look like? They're called synapses. And here's a bunch of Google images results for word synapse. So as you can see, there is now an archetypical image of a synapse as well, kind of similar to this archetypical neuron that we've seen in textbooks, except this one is much younger, of course. So they are usually depicted as these sci-fi entities hanging in the void and transmitting some glowing something from one part to another. And so there is some truth in it, in the sense that, I mean, they don't glow, of course, in real life, but the main message that artists, all these CGI artists seem to have captured is that there are two parts that meet each other. So synapse comes from the Greek conjunction, and the idea is that it is kind of one entity. It is important for how our brains are built, but at the same time, two different parts of the synapse come from two different neurons. So every time we see these parts of a synapse, these two elements, two halves, come from different cells. And the cell bodies may be located on the opposite sides of the organism, right? Of the body. For example, in case of a neuron from your brain talking to a neuron in your spinal cord in order to make your hand move. Here's a more realistic picture. Here we don't see how individual synapses look like, but we can see where they are. So here the cell is glowing green because scientists made it glow green. They artificially tricked it into producing some green glowing thingy. So it's glowing green, and synapses that are on the cell are glowing yellow. And you can see that pretty much any dendrite that the cell has is covered with synapses, with hundreds of those. So each cell receives thousands of connections from other cells. Here's a close-up with a yet slightly different color scheme. Here the cell is red and synapses are green. And you can see that they are not quite exactly on the surface of this protrusion, but they're kind of nearby, and sometimes you can see this thin neck leading to the synapse. And here we only see, I guess, postsynaptic parts of synapses. So we don't see the cell that's sending signals here. We only see the parts that receive signals, because if we try to visualize everything, obviously it would have been a mess with thousands and thousands of fibers. But you can imagine the part that you don't see, right? So synapses were actually, like, for a while people didn't realize that they even exist, because when people realized in mid-19th century that brain mostly runs on these electrical signals, on action potentials, and imagine that they even managed to use some weird, really steampunk technology to register, to detect action potentials back in late 19th century. So they realized that neurons have this funny shape, and they realized that there are electrical signals going on in the brain. And the major concept was that the brain is this one large network where cells are directly fused and connected to each other. Then neurons were discovered. People grew more accustomed to the idea that there are individual cells, but still it felt logical. If there are electrical signals, then probably the connections between cells are also electrical, right? And so there were two camps: those people who believed that synapses are electrical and those people who believed that synapses are chemical, and they fought for a while. Around 1920s, the chemical theory won. Otto Loewi was a Jewish scientist who worked in Austria and had to later run from Nazis to United Kingdom and to the United States, and he got a Nobel Prize for his discovery. And the discovery was this peculiar thing that he had a frog heart in a tube, and the thing with frogs is that they're cold-blooded, right? So if you take a heart out of a frog, because the metabolism is slower, it keeps beating for longer. Actually, even a human heart keeps beating for quite a while, but with a frog heart, an isolated heart keeps beating for hours. And so he had a heart like that, and then he stimulated some nerves, and he noticed that the heart slowed down. This on itself doesn't prove anything, right? So there is clearly some communication between the nerves and the heart, but whether it's chemical or electrical is still unclear. However, what he did then, he took the fluid from this heart and added it to a different heart, and it also slowed down, which meant that there is something in the fluid, there is some chemical in the fluid, which suggested that the connection is chemical. And from this very indirect evidence, gradually, we realize that connections are chemical. In a way, it is counterintuitive, right? Because cells use electrical communication within the cell, but then for some reason switch to this chemical mode in between. Why? An even bigger surprise happened in the 50s. So in the 50s, people tried to figure out how the chemical communication even works. So okay, some chemical is involved, but how is it released? And what's perhaps especially interesting, this communication has to be fast, right? Because, nervous system needs to be fast. In a way, nervous system appeared first in order not to be smart. Eventually it became smart, but originally it appeared to be fast, because if you just need to communicate from one part of the body to another part of the body, you could also use chemicals. You can just release chemicals in the blood or whatever fluid that's pumping and change the way you move, except that it will be very slow. So originally nervous system developed in order to enable fast communication between different parts of the body, so that those primitive animals that are long gone wouldn't get eaten by other primitive animals. But if communication from a neural cell to another neural cell, or in this case to a muscle, if it's based on a release of a chemical, how can it be fast? And yet it needs to be fast so that an animal could, say, jump away. And in these early experiments were made in frog muscles, because again, you can work with a frog muscle for a long time. But frogs, they sit immobile for a while maybe, but then at some point they jump, and it's a fast process, because they probably are trying to escape either from you stepping on it or a bird eating it or something like that. So it's a matter of survival. It needs to be fast. How can you release so much chemical so fast? In the 50s, people invented fancy electronic devices. And that's kind of interesting how one part of science can only progress once another completely unrelated parts of science has its share of progress. The war happened and people developed all these electronics and amplifiers. And now suddenly these amplifiers could be used not only to amplify sound, but also to amplify electrical signals from biological preparations. And so people used amplifiers and invented these tiny electrodes and put electrodes in the muscle near to where an axon from a neuron comes to the muscle. And here's what they recorded. So if you record the potential or a current, if you record the electrical activity of a muscle, you will see that there is some noise and the muscle sits at a certain level, so something happens, but not much. But then every now and then you have these events, you have these things that happen again and again. See, I'll mark 3 of those, but you can see that every trace has lots of them. So it's like nothing, but then suddenly pew, 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 pew. And what could this be? So these are not action potentials in the muscle, because when action potential in the muscle happens, the muscle contracts. These events are much smaller in amplitude. If you have lots of those, maybe you will eventually reach an action potential for the muscle, but these events are about, say, 100 times smaller in amplitude than what would make the muscle contract. So these are not action potentials in the muscle. Then another thing, you might think maybe these are action potentials in the fiber that comes to the muscle. So maybe the neuron spikes, the neuron that connects, the neuron that sends information to the muscle saying like contract, it's time to jump. Maybe this neuron spikes, and every time it spikes you see these events. But actually that's also not true, because in this preparation the neuron was gone. the muscle was cut out, and while the very last few millimeters, micrometers of the axon still remained in the preparation, it was not spiking. So the very connection from the neuron to the muscle was intact, but the neuron was gone and it wasn't spiking. It's something about the connection itself that created these discrete events. It was unexpected. Then, also in the 50s, electron microscopes were perfected. They were invented a little bit before that, but they were perfected, and people were able to make electron microphotographs. And so they made first electron microphotographs of a synapse, and here's what they saw. And so if you combine these two pieces of evidence, that an electrical recording from the postsynaptic cell, in this case a neuron, is full of these events, but also that the presynaptic cell on electron microphotograph looks like that. So there are events and then there is an electron microphotograph. What does it tell us about how the synapse functions? What is the discovery that people made? Make a guess. Pause the video, make a guess. I think you probably figured it out, right? There are events, so time goes like that, and every now and then something happens, but also if we look in the synapse, there are lots of somethings here. There are lots of little bubbles. And so these bubbles are sitting in the presynapse. They seem to be crowded at the part where we have a connection to the other cell, because see this black matter, this dark stripe, so that's where the presynapse is actually connecting to the postsynapse. So there are bubbles and there are events, and it's logical to assume that maybe these events come from these bubbles. And it's kind of nice, but also kind of counterintuitive. It's counterintuitive because, again, it feels weird that you have an action potential in the presynaptic cell, you eventually have an action potential in the postsynaptic cell, even in case of a muscle, but especially if you're looking at a synapse that connects to neurons. So it's electrical language before and electrical language after, but between the cells, we have a chemical synapse. And moreover, this chemical synapse happens to involve these little bubbles. You can kind of relate to this bubble thing from the speed point of view, because if the presynapse just had to sip chemicals, just had to transport them across the membrane one molecule at a time, it would have been, of course, incredibly slow. So one thing that we can learn from these events is that you can have an entire bubble released pretty much instantaneously, which is good news. It means that synaptic connection can be fast, but still it's kind of weird. It's like, it feels that we go from a fast process, which is electrical communication, to a slow process that is chemical communication, but then we happen to make chemical communication faster by using these bubbles. Interesting. There are a few words, special terminology that we should mention here. One is the word vesicles, which is a weird word, and it is just a word for these tiny bubbles. For some reason, specifically for these little bubbles in the presynapse, usually people call them vesicles, presynaptic vesicles. And it is just a habit that people have. By the way, what are these bubbles made of? Like, what makes the wall? What do you think? If you thought that is the same thing that made any other wall, including the cell wall and the nuclear wall, the membrane, you're right, it's made of membrane. So these vesicles are made of little pieces of membrane, and then they are produced somewhere in the presynapse, and then they're crowded near those parts where they can be released. And here we can see several schematic representations of several different synapses, and the idea that this image tries to communicate is that different synapses can have very different shapes. They can be large, they can be small, they can have huge presynaptic part and tiny postsynaptic part, or the other way around. There are lots of different types, but what's common is that there are two parts, presynapse and postsynapse, and vesicles are produced by the presynapse. And then what separates them is called a synaptic cleft. So this part in between, this small gap in between, is called a synaptic cleft. And vesicles crowd near the synaptic cleft, and at some point they release their content into the synaptic cleft. And sometimes it happens spontaneously. And that's, again, how these recordings look like. This particular connection from a neuron to the muscle needs to be fast, and it's so fast that vesicles are sitting as close to the membrane as possible. In fact, they're almost fused. Like the name for that is docked. So if this is the cleft, the vesicles are sitting right there, very like as ready to be released as it gets. They're filled with stuff and they're ready to be released. And every now and then they just get released spontaneously because they just released by mistake. But actually they're waiting for the signal to come, for the action potential to come from the presynaptic neuron, so that this process would be triggered and then they would be released. And here's the picture of a neuromuscular junction, which is this very specific type of a synapse between a neuron and the muscle, where we can see vesicles and we can see them docked. We can see them already placed on the membrane, ready to be released. And every now and then they fuse and stuff spills out. And if the action potential comes, all of them get fused together, but sometimes kind of false starts happen and they get fused just because, you know, they're so ready and they would be like, oops, sorry, didn't mean to do that. But so next question. Okay, chemical got released from the vesicle and into the synaptic cleft. Now we have chemical in the synaptic cleft. We did half of what needs to happen, right? So you have the presynapse, it has vesicles, they are prepared, and then there is the postsynapse, and it's waiting. Vesicles fused with the membrane, the neurotransmitter got into the synaptic cleft, and it's now in the synaptic cleft. What next? What's next? Because now the postsynapse should somehow feel it. How can it sense the presence of a neurotransmitter? Any guesses? Make a guess. What can sense the presence of the neurotransmitter? Okay. Channels. We talked about channels. Remember what they're made of? Proteins. So fancy proteins that come together, that sit in the membrane, that create this pore through which ions can go, and they can be closed or they can be open, and there may be different things that make them closed or open. So we talked about stretch activated channels and we talked about voltage activated channels. But in this case, we have a chemical in the synaptic cleft. Can there be chemical activated channels? Indeed, the answer is yes. And they're called receptors, which is kind of weird because receptor typically is a protein that can sense the presence of a certain chemical. But so this aspect of functioning, the fact that there is a certain chemical, in this case a neurotransmitter, and it can bind to a protein and something changes about the protein, this is called a receptor. But this receptor is also a channel. And so the name for that is ionotropic receptor, which is really fancy, but basically what it means is that this is a receptor that can allow ions through. Ionotropic receptor. One protein plays two different roles, have two different functions. It is a channel. It can let stuff through, as we can see here, but it's also a receptor. It can sense the presence of a chemical. And so that's one way synaptic transmission can happen. There may be these receptors, ionotropic receptors in the postsynapse, and they would sense the presence of a chemical in the synaptic cleft, open, act as channels, and let ions through. And then something interesting will happen in the postsynaptic cell now, right? There is also another option, and it's called indirect transmitter action or metabotropic receptors. So in this case, the receptor is not a channel. The receptor is just a receptor. it catches at some point the neurotransmitter, it senses it, but it is not a channel. The channel is sitting separately somewhere in different part. And the way they communicate is via other chemicals called intracellular messengers. So what happens is the receptor senses the presence of a chemical. Some chemical reactions happen inside the cell. This giant molecule may interact with another giant molecule that would interact with another giant molecule, and eventually they change something about the ion channel that's sitting elsewhere. I mean, in the same membrane, in the same cell, and it starts to let ion through. Two different ways of how the postsynapse can react to the presence of a neurotransmitter in the synaptic cleft. Direct and indirect. Now some words. Again, lots of words that are involved. Receptor. In this case, a protein that can sense the presence of a chemical, a neurotransmitter. And by sense, it means that they combine, so it catches it. It has a special part that is kind of ready to accept the neurotransmitter, that has the shape that matches the shape of a molecule of the neurotransmitter. And so when it gets in, it's like the key and lock interaction. So when it gets in, something happens. Next word, agonist, also called ligand. So this is a fancy name for neurotransmitter. In this case, agonist is a neurotransmitter. Ligand is something that binds, from Latin to bind, I think. But both words mean the chemical that is ready to bind to the receptor. And sometimes, like if you're talking about how a synapse works naturally, this agonist will be produced by the body, so it will be cool. But you can imagine also that sometimes some animals create weird, or plants create weird chemicals that disrupt this process also by serving as agonists. Because remember, poisons and venoms and stuff like that. So there may be natural agonists that occur in your body naturally, but there may also be unnatural agonists that were chemically synthesized either by an evil plant or by an evil mollusk or something, or maybe by a really nice medical researcher who is trying to help you, right? So all of them, whatever binds to the receptor and changes its function and creates all these downstream effects, would be called an agonist. Okay, channel. Channel, we use the word channel. So the key thing here is sometimes channel is also a receptor, and sometimes channel is separate and receptor is separate, and they talk to each other. Second messenger, those messengers in the cell. Why are they called second messenger? Who is the first messenger here? I'm guessing the neurotransmitter is the first one, right? So if you just say the messenger, people may think that that's a transmitter. So for anything that happens after the transmitter was released in the cleft and caught by the receptor, for all this process in the cell, we use the word second messenger, even, if actually it's more than one. So it would be third or fourth, but I don't think people use third or fourth. They call all of them second messengers. Co-agonist and antagonist. Okay, so if you understood the word agonist really well, so something that binds and causes the consequences, then we can figure out what a co-agonist and what an antagonist mean. Co-agonist, well, let's start with antagonist. Antagonist, what is an antagonist? Anti means against, right? So it has to be something that opposite of what happens here. So for example, another chemical that like blocks this part. So now the poor little agonist cannot bind. And again, it may be a poison, or it can be a really nice chemical that we use medically, medicinally. We'll talk about that a little bit in the future. And co-agonist is like a friend of the agonist, right? And sometimes it happens in the body naturally. Sometimes, again, it can have medical relevance. But we can have chemicals that on their own don't cause this reaction to go, but they kind of help facilitate this process of binding. Maybe they like bind in a different place and make the channel or the receptor a little bit more eager to act. In this, maybe they have to be present, maybe they're optional, so everything is possible, but co-agonist means that. Then a funny thing, ionotropic and metabotropic receptors and also ligand-gated channels. So it just so happens, because these proteins are both receptors and channels, that you can talk about them as a receptor that also happens to be a channel, or as a channel that also happens to be a receptor. If you follow this line of thought that it's a receptor first, but it's also a channel, then you call it an ionotropic receptor. So the receptor that can let ions through. But you can also kind of start from the channel part, and you can say, okay, that's a channel, but it's also sensitive to chemicals. And in this case, you will call it a ligand-gated channel. But ligand-gated channel and an ionotropic receptor are exactly two same things. It's like saying, you know, doctor who is also a climber, or a climber who is also a doctor. Still same person. All right, next. Yeah, so now as we described that, a logical question is why? Why do we have this complexity? Why are there two different ways? Like, both of them solve the problem. Why wouldn't there be only one? Why do we have to study that, basically? Why do we have to deal with that? So let's think about that, and let's think about two questions. So first, if you look at these two approaches, so in one case, the receptor happens to be a channel, and in the other case, the receptor is separate and channel is separate. Which one is probably faster? Make your bets. Is direct faster or is indirect faster? I won't even use the ukulele part. You probably assumed correctly that direct is faster because you have only one entity here, right? So obviously having one entity is faster than having like all this not middlemen in this case, but middle proteins, middle cascades. Indirect transmitter action would naturally be slower, at least all other things being equal. But then why the slower part still exists? So if this is fast and you can just create channels, and they exist, there are so many of them, channels that are directly activated, that can be opened by a chemical. If this is available, if this kind of technology is available, why wouldn't through mutations cell gradually switch from this one to this one? Like the cell would have a little bit more of that, a little bit less of that, and if it works better, if it's cheaper, if it's easier to organize, if it's faster, they would escape faster, they would like forage, eat food faster, right, these animals, and they would have more babies, and gradually babies with a superior technology should win. But what we see is that both of them exist, and it's not like this slower one is rare. Nope, it's everywhere. Why? What's cool about slower one then? In which aspects, here's the question, in which aspects may this thing, the slower process, still be superior? Are there any upsides to the indirect transmitter action? Pause the video, think. This is a hard question, so think, write it down, make your bets, maybe write two, three different ideas here. Why? Why does it exist? Usually when I ask this question in class, students come up with basically two reasons. So I don't know which one came to your mind, but let me, like, hopefully you hit at least one of those, but maybe two. First, look at that. We have this intermediate element here. So you caught a molecule, it affected the metabotropic receptor, and it created an intermediate second messenger. And now this intermediate second messenger can walk around and like change something about how the cell functions. Can it be that actually one intermediate second messenger, one intracellular second messenger, can affect more than one ion channel? Or maybe the metabotropic receptor can generate more than one intracellular second messenger. So it feels like here everything is like one-to-one, one molecule, one channel. But here, this assumption may not necessarily be true, right? One molecule can cause more consequences if only you either have more than one intracellular messenger generated, or this intracellular messenger affects more than one ion channel. And indeed, that's what happens. We can have an amplification cascade. So one molecule caught would cause several, like one molecule caught by the receptor, I will use this really schematic drawing here, can cause several intracellular messengers. Okay, like messengers. These are molecules probably, but I will draw stars because like we have stars here. And then each of them can go and affect different receptors. And in fact, I mean different ion channels, sorry. But in fact, they don't even have to be different ion channels. Maybe some of them will go to the nucleus and affect how the which genes are expressed. Maybe they will affect other parts of the cell functioning as well. So this we can have cascades, we can have amplification cascades where small original message which will cause bigger consequences. Right? So that's one benefit. And the other benefit is that it feels like if you needed to fine-tune something, this second option, the indirect option, seems to be inherently more fine-tunable. So if it happens so that you may have to like fiddle a little bit with the way how the synapse works, it just feels like there is more potential here for subtle effects. Right, like the fact that I noticed that that I just mentioned that maybe these second messengers affect not one but two different processes. Only one of those is about ion channels, and the other one is completely unrelated and related to how the cell grows or wanes or adds stuff or removes stuff, right? And if you look at actual cascades for metabotropic receptors, they are incredibly complex. And this diagram here, like this diagram here, is still super simplified. And you're not supposed to remember it. The reason I'm showing it is that I just want to show you the level of actual complexity. Even if we look at one type of a cascade only, here's what happens. Look at how many different players are there. So I rewrote it as this weird paragraph below, just replacing all the terms with the word thingy. And what happened, what I got is that a metabotropic receptor lets go of a large thingy that's usually parked there. The large thingy immediately breaks into several smaller thingies, catches a tiny thingy, breaks into mid-sized thingies, and then these mid-sized thingies start walking around, bumping into large thingies that catch other type of small thingies, convert them into other types of small thingies that then they diffuse all over the cell and affect various proteins and various, like, all aspects of cell life, essentially. You get the point. If you want the presence of a neurotransmitter in the cleft to have big consequences for the cell, this is a much nicer approach. All right. So far we talked about synapses as a way for one cell to communicate with another cell, right? Which means that what happens probably is that we have one cell, we have another cell, so the presynapse and the postsynapse, and the action potential comes here, and there are vesicles, and these vesicles are fused with the membrane, and what should happen eventually is that an action potential should happen in the postsynaptic cell, and the action potential should continue moving in this direction. So the action potential should somehow jump from one cell to another, but not directly, but with the help of this chemical synapse. And this is called excitatory synapse. And most synapses in our brain are excitatory. As you can guess, usually when one neural cell is talking to another neural cell, that's because it wants the signal to go further. Not all synapses are excitatory. Some of them either change things a little bit and they're called modulatory, or are inhibitory and tell other cells to shut up, which is called inhibition. But the majority of synapses are excitatory. So one question that we need to ask is, when we have this excitatory transmission, what ions would go through these channels? Like the channel opens for one reason or another. What ion would go through the channel? Most probably. Like what ion would you send through the channel in order for an action potential in the postsynaptic cell to appear? What ion goes through the channel? Make your bet. What ion goes through the channel so that the action potential would happen in the postsynaptic cell? So let's think, which ion really wants to depolarize the cell? Can we just use action potential as an example? In action potential, which ion depolarizes the cell? Sodium. Okay, and sodium is outside. So if you make these channels that are normally closed, when they open, if they make them permeable for sodium, it will do the trick. And that's indeed the case. In excitatory synapses, channels that sense the presence of the neurotransmitter are mostly permeable for sodium. Good. So next question, what neurotransmitters are excitatory? The main excitatory neurotransmitter is glutamate. And about 80% of synapses in our brain are using glutamate, or a fancy word for that is glutamatergic. So they use glutamate, and that's the most common neurotransmitter in our nervous system, because most synapses are excitatory, and glutamate is the key excitatory neurotransmitter. So it's like the working horse of our nervous system. Glutamate is chemically a basic amino acid, so we have it in our proteins as well. So it is used as a brick, as a building block to make our proteins. It's a tiny amino acid, one that's cheap to produce. But also these bricks are thrown by one neuron to another as a means of communication. Incidentally, it means that eating glutamate, eating monosodium glutamate, is not risky because we're full of it, essentially. It's not exactly like water, but it's somewhere there. It's closer to water than to vitamins, for example. It's a chemical that is very abundant and we can synthesize it. So it's impossible to overdose on MSG, on monosodium glutamate. And there is no reason to be afraid of monosodium glutamate in the food. We'll talk a little bit more about that when we talk about taste. So that's the most common excitatory neurotransmitter. What else do we have? That stuff in the neuromuscular junction, so the synapses, special synapses on the muscle, where a neuron is connected to the muscle, they don't actually use glutamate in us, in you, and in me. They use a different chemical called acetylcholine. And this is probably the second most common excitatory neurotransmitter in our body. Not in our brain, but in our body. So glutamate, the most common excitatory neurotransmitter in the brain. Acetylcholine, the most common excitatory neurotransmitter in the body. I mean, in the body that's not the brain, I guess. Two footnotes here. One footnote, which I find really cool and weird, is that in us, glutamate is in the brain, excitatory. Acetylcholine is excitatory in the periphery. In insects, for example, or in crustaceans, or in most invertebrates, for that matter, it's kind of reversed. So we use glutamate to think and acetylcholine to move, and they use acetylcholine to think and glutamate to move, which doesn't make... So this is not one of those questions where I can do the ping and make you think why it is so, because there is no answer. It is just so. And we can kind of infer that it is just so, that it's a random not even choice, but random thing that happened during evolution. We can figure it out if we compare a bee to a mouse, because bees are smart, but they think with acetylcholine and move with glutamate, while mice are also smart, but they think with glutamate and make their muscles move with acetylcholine, which kind of suggests that this doesn't matter. It could have been the other way around, and maybe in some alternative universe there is some alternative Earth where it's swapped. Not everything that we see in biology can be explained by reverse engineering. Sometimes the answer is just, yep, just so. Why do we have 5 fingers? Why not 4? Yeah, could we have 4? Could we have 6? Yeah, probably. But we happen to have 5. Okay, not a big deal. Another footnote. Acetylcholine, and that's an important one, kind of, if you want to, especially if you're interested in chemistry and pharmacology and different sorts of, how chemicals affect how your body work. Acetylcholine is not always excitatory. So glutamate is almost always excitatory. With acetylcholine, it's complicated. It's always excitatory in the neuromuscular junction, but elsewhere in the brain, it can be either. And even in the peripheral nervous system, it can be either. For example, that stuff that Otto Loewi found that slow the heart down, it turns out that what slows your heart down is also acetylcholine. So while it's excitatory in the muscles, when an entirely different cell pours a little bit of it on your heart, it slows down. And that happens, for example, if you ate lots of hamburgers or sushi and now you're like lying on your couch, relaxing, and your heart slows down and you feel sleepy. This process, this regulation is actually mediated by acetylcholine, it turns out. But we'll talk about that later as well. It's an entirely different topic of how your brain talks to your internal organs. Now, let's do the sense check. What will happen if you block postsynaptic receptors? What are these postsynaptic receptors again? Presynapse vesicles and neurotransmitter gets released, so vesicles open, emptying the synaptic cleft. There are postsynaptic receptors sitting over here. Can we block them? The neurotransmitter acts as an agonist. Can there be an antagonist? And what will happen if we block them? For example, if we look at acetylcholine in the neuromuscular junction. This is a neuromuscular junction, this giant synapse on your muscle. If you block all postsynaptic receptors in a neuromuscular junction, what will happen? Make a bet. If you block all postsynaptic receptors, vesicles will still be filled with neurotransmitter. They will still be docked, ready for release. They will still be released. Neurotransmitter will still go to the synaptic cleft. But then, if the receptors are blocked, they won't be able to sense it. They won't be able to bind with it. Or maybe they will try to, but nothing will happen. So there will be no transmission. And if this synapse goes from a neuron to a muscle, then the muscle won't hear the signal. So the muscle won't depolarize and the muscle won't generate an action potential, which means that it won't contract, which means that the animal will be paralyzed. And indeed, that's how curare works. You might have heard about curare because it sounds very grim, dark, romantic, and it's used in blowguns because what it does, if you make a dart and cover it with curare that comes from a plant, right? And you send it to a deer, then deer gets paralyzed. It doesn't die. And in fact, if it's just slightly paralyzed, it just cannot run, so you can catch it, but it doesn't die on you because the muscles that make it breathe are a little bit more resistant, so it will probably be able to barely breathe, but it won't be able to run. And so you can bring this deer to your village. And if you don't have a refrigerator, that's a really nice way, gruesome, but nice way to preserve meat by having a live deer. And then you cook it. And the cool thing about curare is that it breaks down if you cook it. So even though it's poison, you can eat this deer later. That's how antagonist for neuromuscular junctions became famous. But now scientists use it, obviously, because as I mentioned before, any way of manipulating experimentally the way neural cells work will probably be useful for one experiment or another. So it's totally used in neuroscience experiments, and you can order it from like tiny amount, not enough to kill a deer, but a tiny amount to add to your bath solution where you have some neural cells, and block acetylcholine receptors if you need to. Now, another question. So it may sound technical at first, but it's also related to actual real stuff. What happens to the neurotransmitter in the cleft once the transmission is over? Presynapse, postsynapse, vesicles get released, bind to the receptor. Okay, good. But what happened to the neurotransmitter? Here you can use your common sense to come up with about 3 or 4 different things that can happen to it. Because it cannot stay, right? If it stays, then the concentration will just increase, and at some point there is no reason to release more because the cleft is already full of it. So it cannot stay. Something should happen to it. If we want to use the synapse again, we should clean up. So what can happen? What can be behind this process? Make a bet. All right, what can happen? Basically, I think we can come up with 4 different answers. Passive diffusion. So you just have a cleft and it's slightly open on the sides and everything just seeps out. And if you wait enough, it will just kind of diffuse out because chaos wins over order. And so the concentration will drop just because it will kind of get dissolved in all the fluid that surrounds the cells. And that's an option for some synapses, definitely. But probably not for synapses that need to be fast and may have to be reused at a high frequency, work, then be silent, then work again in a matter of only a few milliseconds, or for synapses that release so much neurotransmitter that relying on diffusion would not work anymore. So what can be done as well? You can have reuptake of the neurotransmitter by neurons. So the neurotransmitter is released fast, in a fast way from the vesicles, but then there are transporters and slowly, much more slowly, neurotransmitter is moved back into the neuron because eventually what you want to do is to pack it into vesicles, into new vesicles. You need to prepare new bubbles and prepare them for the next wave of activation. And that's definitely an opportunity, an option. Another, reuptake by astrocytes, and that's what happens with glutamate. So when I showed you pictures of glia, of glial cells, of those cells in the brain that are not neurons, and we have lots of those, I told you that neurons are lazy. Neurons really want everything to be done for them. They are these highly specialized cells, kind of arrogant. I imagine them being arrogant. And among other things, in glutamatergic synapses, again, 80% of synapses in our head, most of glutamate is actually reuptaken not by neurons, but by astrocytes. And then they prepare it and give it to neurons like in a nice package so that they could pack them back into vesicles. And the final approach would be to break it down. And that's the case for acetylcholine. Just the molecule for acetylcholine is larger and it's easier, I guess, to break it into 2 molecules and then later synthesize again. And so in our neuromuscular junctions, in order to depolarize the muscle, we release lots of neurotransmitter, but we want it to be cleared fast because otherwise we'll have a spasm. We'll just activate our muscles and stay like that. But we want, to be able to do fast movements. So to do fast movements, we need to be able to send the signal and then not send it. And what happens is that it's broken down by special enzymes, another type of proteins, so that the concentration of acetylcholine in the synaptic cleft would fall down real fast. And then it's also reuptaken and resynthesized and prepared for the next wave of excitation. And here are 2 ways in which it actually relates to normal human life. One, you might have heard, and by that I mean you definitely probably heard, about Prozac, which is the most common, or maybe not the most common, but one of the early antidepressants. And what it does, it decreases reuptake of serotonin. So it's called an SSRI, selective serotonin reuptake inhibitor, which means that it inhibits reuptake. So it decreases reuptake. It decreases something that decreases the concentration of neurotransmitter in the cleft, which means that it stays for longer in the cleft. And somehow, in some people at least, in some forms of depression at least, it may help. And people feel less depressed. Actually, how it works is kind of unclear, and we'll talk later about that. But at the very least, what's important is that messing with this process may be one of the ways we can affect the brain. So we can try to add chemicals that make channels or receptors more active or less active. But another option, we can mess with the speed at which neurotransmitter is degraded or removed. And another example is treatment for myasthenia gravis. Myasthenia gravis is a relatively common autoimmune disease in which the body attacks its own acetylcholine receptors for acetylcholine in the neuromuscular junctions, which means that you have less and less receptors in your neuromuscular junctions, and neurons cannot talk to the muscle that effectively. And so people feel weak, and it's hard for them to move, it's hard for them to swallow, it's hard to keep their eyes open. And one of the treatments, at least symptomatic treatments, is giving people a chemical that decreases the breakdown of acetylcholine. And again, what happens is that acetylcholine stays for longer, which means that faster movements will be a problem, but at least for slower movements, the intensity of this movement is recovered, because then people have fewer receptors but more acetylcholine. Finally, here's the weird part. Electrical synapses actually do exist. Remember those fights of people who believed that interaction of neurons should be electrical, and those who claim that, like prophetically claim that it would be chemical? It turns out that electrical synapses do exist, and sometimes two different neurons are just connected with these special channels that don't open and don't close. They're always open. They like just bind them directly. And then when one neuron spikes, here's a spike in one neuron, another neuron just automatically also spikes. It's like a hard connection of two neurons into one, almost like one cell, in the sense that, I mean, there are still different cells, but the cytoplasms, the internal media, is now continuous because they are connected through these channels, and everything, like even small molecules, can go through these channels. So electrical synapses do exist. Why do we use these pesky chemical synapses then? Why the majority of our synapses are chemical? Why electrical synapses are relatively rare? What's cool about chemical synapses? Like, why, again, we go back to this reverse engineering nature. Why would you go from the electrical language to chemical language only to get electrical language again? Why wouldn't you just connect two cells and be done with that? What do you think? Make your bet. I hope you notice that this question is kind of similar to a different question we had before. For chemical synapses, why there is a direct and indirect pathway. And same like there. So indirect was slower, but also more malleable, like more tunable, fancier, could have many different consequences, and so on. My guess is that we can say similar things about chemical transmission. Of course, we can connect to cells, but then imagine if you need to make the cells less connected or more connected. Of course, you can change the number of these connections, but it's a slow process. What if you need to suddenly inactivate this connection? Like maybe you're a frog who jumped, but then now you realize that like the place where you're sitting under the leaf is the only safe place in the entire backyard or something. And so you definitely don't want to jump again, right? Lots of what we do works like that. We need it one moment, but then we don't need it at the next moment, but then we will need it again. So we need to be able to switch things on and off really effectively and efficiently if we want to survive. There may be situations when like some connections should be always on, but for most connections, we should be able to quickly change something about it. And it's clear with electrical synapses, even from this diagram, like from the looks of this diagram, you can imagine that, okay, this is something that can be used to bind to cells for a relatively long period of time, but it's not something that can be turned on and off on a whim. While with chemical synapses, with all the agonists, antagonists, even with direct transmission, when we have ionotropic receptors, even with them, we can tune it so well. We can like block the receptor from the inside or modulate it or whatever. we can change the way stuff is released from the presynapse, right? Lots of different things to play with. While here, it's kind of hardwired and that's it. So electrical synapses exist, but they're not too widespread. Okay, now we go to a slightly different presentation. It's related to everything we talked about. It's related to the action potentials, and it's related to synapses, and actually to electrical synapses, as it will turn out. But it's also kind of almost like an application of everything we talked about before. And it's actually about shrimp and escape responses in shrimp. But the way I think about it is a presentation that shows what can one neuron do. So how useful can one neuron be? One neuron that can either spike or not spike. Let's see. And we kind of talked about that already when we talked about squid and giant axons, but here we have a shrimp. So what's cool about shrimp? I mean, lots of things are cool about shrimp, but one cool thing is that they can escape, as many other small animals. And depending on where you touch them, they can escape forward or backwards. And the way they do it, they have this telson, the tail, the special tail. And if they flip their tail, they will go in the opposite direction. And if you touch a shrimp close to its head, what happens is that the shrimp, this is supposed to be shrimp, this is the eye of a shrimp, these are all the legs, and this is the tail. So the tail gets curled, and because of that, water gets ejected from here, and the shrimp goes over there really, really fast. Okay, so if you touch the shrimp around near its head, it goes in this direction. If you touch it at its tail, as it turns out, it also bends the tail, but in a different way that bends the shrimp kind of upwards, which makes the water shoot down and makes the shrimp escape forward. So it has this escape maneuver, which is actually kind of two different maneuvers. And both are very fast, and both involve bending the tail, but the tail is bent in two different ways, depending on whether it needs to escape forward or backwards. And yeah, it results in two different escape maneuvers. And it's so fast that it's hard to see, and people weren't even sure how it worked until they were able to, make a video of it and look at it frame by frame. If you ever try to catch a shrimp in the ocean, which is an activity I totally endorse and recommend, go to some place with tidal pools and try to catch a shrimp that got like stuck in a tidal pool. They are insanely fast. So how do they sense the touch? Let's just talk about this network step by step. How can they sense that they were touched? Well, they're shrimp. They have armor, right? So unlike us, they cannot rely on a bendy skin. Remember our touch receptors and our stretch activated channels? In shrimp, it's slightly different. They have tiny hairs that stick out of their body. So the body is solid, but then there are these sensory barbs. And when they bend here or there are some neurons that are sitting somewhere in the basement of these barbs. And as the barb bends one direction or another, it touches these sensory endings and some neurons spike. They spike in response to a touch. So we have the sensory part. The shrimp can actually sense that you touched it. Where do these neurons send information? So we need to know two things about the shrimp. One, it is segmented. It has these segments. If you look at it, it looks like here how it looks like. It has these segments, and each segment comes with a pair of legs, and each segment comes with some muscles inside, and each segment can articulate, so it can move a little bit. That's one important part for this story, because that's how the tail will bend. And if you bend it more these segments, then the shrimp will go forward. While if you bend these segments, or maybe all segments, then the shrimp won't go up, but instead it will go backwards. Another interesting thing is that there are giant axons. We use this combination of words in an entirely different animal, in a squid. Squid has giant axons, also used for escape maneuvers, but shrimp and decapods, so like lobsters and crayfish and all these guys, they have giant axons too. And what's interesting is that there are two giant axons, lateral and medial they're called. So this is the animal and this is the nervous system. As you can see, yeah, the brain is tiny. Remember, this talks about the brain. Yeah, a little bit more stuff in the head, but not much. And then there is stuff in the middle of the body. Actually, it almost feels like the middle of the body is smarter than the head. Yeah, I guess. But then, so if you look at these connections, so all of these ganglia, all of these neural centers are on a chain, and there are lots of tiny axons here that connect neurons within the chain. But on top of all these tiny axons, there are two huge ones, medial giant and lateral giant. So all the axons are tiny, but then this one is one giant axon that is comparable in size in terms of its cross section to the size of a giant axon in a squid. Because remember, they don't have myelination, they haven't invented myelin. So when they want something that conducts signals really fast, want here again is meant in the evolutionary sense. When it's evolutionary critical to have fast conductance speeds, axons just start to fuse together into this giant super mega cell and giant super mega axon. Another point to consider, electric synapses exist, and indeed if you look at what happens with these giant axons, at every segment there is a ganglion, and there are electrical synapses over here that connect the giant axon to giant motor axon that goes into one of those offshoots. So you have an axon, and it can make, as it runs through the body, it can make these connections to axons that go to the muscles. It's like junctions at roundabouts, where from a giant highway you have roundabouts to smaller highways. And these synapses are electrical, and so whenever they exist, they almost like combine two different cells into functionally one cell. When this one spikes, this one will spike too. All right? So now the question, and this is the question I want you to think about. Take a piece of paper and think for a second. So we have segmented body. We have giant axons that run. We can make connections to those fibers that go to the muscles. Yeah, and we have sensory cells everywhere. So how to make it so that if we touch the crayfish at the back, it escapes forward, but if we touch it at the front, it escapes backwards. Basically, can we reverse engineer what's happening here? What's the smallest, fastest, easiest configuration that will enable that behavior? Think about that. Try to come up with a hypothesis, and then I'll tell you what people actually discovered, how it actually works. Good. So, if you look on this picture, this is actually a wire. So this is from a paper where people try to figure out how it works, and they used wires to record from different cells in the crayfish. And what they figured out is that, yep, there are muscles in every segment, and there are neurons that go to these muscles in every segment. And then there are two giant axons, the medial and the lateral, and just one of them happens to be connected to each and every segment, and another one happens to be connected only to several front segments. Which means that if this one gets activated, the crayfish only bends the front part of their body and escapes upwards and a little bit up front, while if this one gets activated, the medial one, then the entire tail gets curled and the crayfish escapes in the backwards direction. So everything, all that remains is to correctly connect sensory inputs. So when you are touched, when the head is touched, all of them, because you need to escape. So when the head is touched, you need to escape backwards. So you need to flex the entire tail. So the head needs to be connected to this one, while all the sensory stuff in the tail should be connected to this one, right? And indeed, That's what happens. It's just where the connection exists between them and where the connection doesn't exist. That's what defines these two motor programs in this case. And it's a very simple case, but also very typical. It's like a foreshadowing of a huge discussion about how neurons are actually connected. And related to this first question that I posed in the 0th lecture about how you can create complex behaviors out of individual neurons. Well, individual neurons, you can connect them in fancy ways. And the study of how neurons are connected has a special name. It's so important it has a special name called connectomics. It's about connectome is basically a list of which neuron is connected to which neuron. And in this case, this is a very simple connectome. In a tiny worm in C. elegans that has slightly more than 100 neurons, connectome is much more complex already. And we kind of know it, but not quite. We know like big pieces of it. In a human brain, obviously connectome becomes ridiculously hard. And there are people who claim that connectome is really the way the brain should be described. But you can also figure out that maybe like when two neurons are connected, it's not just about whether they're connected or not, but the strength of these connections. So you can argue that this view is a little bit really very simplistic, but at the same time, that's the first step towards understanding how the brain works. One of the possible first steps, figuring out which neurons are connected and which neurons aren't. So that's one learning point from the shrimp study. Another learning point is we figured out what can one neuron do. Even though we have more than one neuron here, they're connected by electrical synapses, so functionally they act as one neuron, just of really weird shape and giant one. And we have two philosophical questions that I ask you to ponder about. I won't be using ukulele. Think about it in your spare time. But this is important. So one, does it look like decision making? Can we use that as a model for decision making? Because the shrimp kind of does make a decision of whether to escape forward or backward. Is it a decision? How to define the word decision, essentially. And another, you probably see where it's going. Does it has a free will? In this example, does it feel like the shrimp has free will when it exercises this behavior, when it uses this behavior to escape? And if it does, how can you tell? And if it doesn't, how can you tell? And as a little bit of a warning, free will is a tricky concept in the sense that it's hard to define. Let's put it that way. I don't want to spoil it, but is it even possible to spoil it? I'm not sure. But free will is really hard to define, so I don't expect you to arrive at some particular answer here. But regardless of whether we in this course can answer it or not, it's a good question to think about. Does the shrimp has a free will here? And how can you tell what the answer is? How would it look like if it had? How would it look like if it hadn't? I think it's a funny question to think about. This concludes our meeting, and good luck.